Hello, I'm Vanessa Sherk from University of Colorado Anschutz Medical Campus. I'm here on behalf of HubLE at the ANZ BMS meeting. I'm here with Eugenie, if you'd like to introduce yourself.
Hi everyone, my name is Eugenie Macfarlane and I'm a third year PhD student at the University of Sydney, based at the Anzac Research Institute in Australia. My wonderful supervisors are Professor Hong Zhou and Professor Marcus Seibel in the Bone Biology Lab. And the focus of my PhD is looking at the role of endogenous glucocorticoids in osteoarthritis and circadian rhythm disruption.
That's fantastic. I hear you had a talk at this meeting. Would you like to tell us what that was about?
Yeah, this year I was really humbled to be nominated for the ANZ BMS Roger Mielik Young Investigator Award. And so in this session, I presented work on how sleep disruption affects osteoarthritis and how endogenous glucocorticoids play a role in regulating this process.
Fantastic. And what were your main findings?
So we used a model of jet lag in mice where we found that chronic sleep disruption accelerated osteoarthritis progression in the mice. So these mice with jet lag had increased cartilage loss, subchondral bone sclerosis, and we also found an increase in synovial mast cell infiltrate compared to mice that just had normal sleep patterns. And then the second part of our study involved looking at the role of glucocorticoids, which are stress hormones secreted by the adrenal glands. The glucocorticoids are known to regulate circadian rhythm, and so we wanted to see if they affected sleep disruption in osteoarthritis. So we used a mouse model where the glucocorticoid receptor was deleted specifically in chondrocytes of the cartilage. And we found that by inhibiting glucocorticoid signaling, these mice were partially protected from the destructive effect of circadian rhythm disruption in the cartilage and bone. So this suggests that endogenous glucocorticoid signaling in chondrocytes is destructive in osteoarthritis and probably plays a role in regulating circadian rhythm disruption in the knee as well.
Wow, that's really striking and very impactful work. So what are your next steps?
Next steps, we've got a lot of work to do still in this area. So we need to look at later time points, looking at late stage osteoarthritis, because this study was in early stage. We also have other mouse models knocking out glucocorticoids in bone cells specifically. So we want to look at the role of bone cells compared to chondrocytes and see if it has similar effects. And we're also interested in looking at the effects of circadian rhythm disruption on bone health in general, because there's some evidence that chronic jet lag can cause bone loss. So we'll see if glucocorticoids can prevent that as well if we knock them out.
That's really exciting work, and I think you certainly have your work cut out in front of you. I think it's going to be fantastic. So congratulations on your talk and good luck with all your work.
Thank you.